name is Srivats.
My name is Chandra.
I'm Sahaj.
And we are from PSBB to present our activity, which is the comparative growth study of Greengram on specific conditions to check the growth. Now, for the next slide, we are talking about the introduction of the study. Now, in this study, we want to see how Greengram, also known as Bannerghatta, grows in specific conditions, and how these conditions could change the growth of Greengram and perhaps other plants. So why do you think we chose Greengram? It's because it's one of the fastest growing plants. It's also a leguminous plant, which we hope this will show us as wide an array of how plants usually grow in these conditions. Now I'll give it to Skanda to explain the objective of this activity.
So the objective is to cooperate and determine how various factors such as soil and environmental conditions affect the plant and the growth patterns. So by analyzing the growth of plants, we want to gain an understanding of how these factors actually affect the plant and what is the best for the plant. And do research.
Yeah, so we've done various observation procedures, and for example, this tomato observation procedure and other various procedures. First, I'll get to the materials utilized. We've used a glass jar, pencils, labels, beakers, markers, 4 varieties of soil, and these 4 varieties of soil are namely black, red, and our sand and cocoa peat. Now over to Srivats.
So I'll explain what soils we use and why do we use them. The first soil that we use is cultivated. This soil that we use is one that specifically retains and absorbs water very well. But the downside of it is that it does not have as much nutrients and it can't give as much nutrients to the plants. So with this we hope to see how well a plant that actually grows in pure cocoa peat soil will be able to grow in the long run. Another soil that we used was black soil. Now this soil is a very clay soil and does contain nutrients, but the problem is that it absorbs water extremely well. So in the humid environment, like Bangalore for example, would this plant in a soil like this grow well? Next soil that we used was red soil. This soil is high in nutrients, especially iron, which is why it gets the red color. But the downside is that it does not have great drainage capabilities, which can lose a lot of water in the long run. And finally, we chose sand. We are an orthodox soil that does not have any nutrients, but perhaps a plant growing this may grow well in the long run. We will have to see. Now I'll pass it on to Sandra.
Yeah, so the nurturing of the Greengram was a very simple process. We nurtured it just how like our moms made it in the kitchen. Basically, we soaked the Greengram for 2 to 3 days, and then after that we put it in a muslin cloth and kept it in a damp corner. We let the animal grow for 1 to 1.5 centimeters approximately, and then after that we pot it in the soil. And over to Skanda.
Okay, for the soil and container, we need to determine how much soil to be added for each container. So we chose about 150 centimeter cube of soil, and we found out that the red soil weighed about 160 grams for 150 centimeter cube. The black soil weighed 156 grams for 150 centimeter cube, and sand weighed 170 grams, and cocoa peat weighed 37 grams. Yeah, passing this on to Srivats for the environment.
So as we know, the environment that a plant develops in might be one of the most important factors for the overall growth of the plant. Which is why we chose 3 environments to show just which environment might be best for the growth of this plant. The first environment that we chose was next to our principal's office, which has a huge garden and is a very open and serene environment. It is very clean and offers plenty of sunlight. But the problem with this environment is that it has a lot of pests, which can lead to stunted growth of the plant. So with this, we can see really how bad pests might kill the plant, or how bad the pests might hurt the growth of the plant. The next environment that we chose was the annex office. This environment that has low humidity and lack of sunlight. It's also a closed space. So we believe this best simulates the urban environment and the city environment. So something like Bangalore, Chennai, or another place like that. With this, we can see just how well green gram grows in an urban environment and how well it can grow in other urban environments and other closed off areas. And finally, we chose an environment near our generator. It's a very polluted environment and it's not exactly green. While it does have more sunlight than the principal's office or the annex office, this pollution might hamper the growth of the plant. So with this, we can really see how bad pollution can stunt the growth of the plant. Next, I'll be passing off this one to Sahash to explain next.
Yeah, so we did a pH test, a normal pH test for all the soils. We have the procedure here. In a liter, 40 ml of water, add 10 grams of the soil into the container, stir the solution for 20 minutes, take the pH paper and dip it into the solution and use color indicator to find the pH. We perform the pH first at the room temperature with the 40 ml of water. After that, we reduce the solution and again we did the pH, and after that only we will see proper results from the pH test. Yeah, now we'll be passing it on to Skanda.
So for the soil testing, we had given it to a biotechnological center, which is at Unicorn or Bio Center. So the results have not yet come, but the devices used by them are the ICP-OES, AAS, and the MEU. So ICP-OES is the inductive coupled plasma optical emission spectrometer, and the AAS is automatic absorption spectrometer, and the MEU is nitrogen emission unit. So basically the ICP-OES, what it does is you are supposed to make a watery solution and put it under it. It will detect all the elements present in it, basically macro and micronutrients. And the AAS is an automatic absorption spectrometer, which basically finds out the heavy metals found in the soil. And we have the MEU, which finds out the nitrogen content in the soil, which we can predict the plant growth variations in. Yeah, passing it on to Shriyas.
Now, of course, in any scientific experiment, you have to have proper procedures to see just how well the experiment will go. Just like that, here we made proper procedures to label each plant and see with utmost clarity how these plants grow. Now, we label the soils in letter code. Black soil we label as A, red soil we label as B, sand we label as C, and finally cocoa pit soil we label as D. And we label each place in number 1. Principal's office we label as 1, annex office we label as 2, and generator we label as 3. And if you're wondering how this all works, you can say that black soil in the generator will be green soil. Now I'll be passing it on to Skanda to do the solution. A growth observation. In this, we just observed the growth of the plant throughout the whole month. Now, we took a letter each growth in each plant and just see how well they do in comparison to other plants and the other environments. The final results of this observation will be given later in the results. Now, I'll be passing it off to Skanda to explain the irrigation.
Okay, so how do we water the plants? So for watering the plants, we checked out the wetness or dryness of the soil, and accordingly we added the water. We measured the water to be precise. We measured it in a beaker, and we used a standard unit as ml. And we have a table for recording all the days of the water we added. Yeah, give it. Okay. So tomato observation, I'm going to give it to the give section.
Yeah, so we wanted to observe these tomato after the 30-day tenure of each plant. So basically it was a very fleshy leaf. We were unable to just tear it and take this tomato out. So we used tape. We put the tape on the back side of the leaf, tore it out, and we were able to get the thin fleshy layer. And that fleshy layer, we put it on a side and observed it under the microscope. That is the basic procedure of how we explain how we observe this tomato. Now I'll pass it over to
Okay, so the pH test results were that black soil, red soil, coco peat and sand were 7, which was neutral in nature, and we found that black soil was 8, which is a weak basic. Yeah, soil is.
As mentioned before, we gave our results to the Biotechnological Center in Mango to get the exact results of the soil testing. Unfortunately, the results have not come yet, but we'll keep you updated on when the results come. Thanks so much for the observation.
So the stomata observation we discerned that the leaf from the generator plant had a darker leaf compared to the principal office. This was probably this potentially indicates the pollution content present in the generator area. Yeah. So basically, due to the chromatic alteration, as I said, this probably indicates the pollution content, and also pollution can affect the stomata. Then stomata is mainly responsible for 3 things, that is gas exchange, water exchange with transpiration, and it also acts as a pivotal point for diseases to get through. So if the weak stomata is there, the diseases might get through. So we found out that the leaves near the generated area was dry, even though we watered it properly. This was probably due to the water loss that the stomata caused, and we also found that there was a disease that got to the plant near the generator. This again, I'm inferring this to the thing that if the stomata is damaged, maybe the diseases can get through easily. And due to also we can see that due to the less gas exchange, we can see that there's lesser photosynthesis taking place, which we can easily see due to the like pale leaves compared to the principal leaves. Yeah, this is the proof. So you can see the stomata is dark and the leaves are quite unhealthy due to the water loss.
This is the growth observation table that we made for how the different soils grow. As you can see, we mark each soil with a color and show the growth pattern throughout different times. With this, you can see the black soil, however, is growing well, seems to be dipping later on, and we can also see that the cocoa peat does not seem to be growing as well, but and that could be attributed to other factors such as the lack of nutrition. The sand seems to be growing comparatively better than the others, but we'll see if this growth trend continues later on. Now, these are the results for the environments that we see. The annex office, which represented the urban environment in a closed-off area, did not make it. It only survived about 13 or 14 days before death. This is perhaps due to the lack of sunlight or the high amounts of humidity here, which caused water stagnation. The generator plants grew well, but they looked much more healthy compared to the other plants, which could be due to the pollution or other factors. And finally, the principal's office grew the best comparatively, but the pests seem to have stagnated the growth slightly. Still, we believe that the principal's office might continue to grow better, and only time will tell on the new results. This is the final graph that we made on the growth of the plants and how in different areas. As you can see, the annex office did not survive long, while the generator grew the fastest, but seems to be going to a dip later. And finally, principal's office has the most steady growth over. Now, for the recommendations, since the biocenter results have yet to come, we can't give you accurate recommendations, but on our experience and what we believe in, cocoa pit, a soil, should only be used as a fertilizer and as a supplement rather than a main soil, which is lack of nutrients. Sand shouldn't be used as much because their growth gets slowed down. Red soil seems to be the most stable, but has high drainage capabilities, which in arid environments might cause damage to the plant. And finally, black soil, since it absorbs so much water, can also cause water stagnation. So it should not be used in humid environments. This is a picture gallery, where you can see how the plants are growing. The first is the principal's office. Second, first is all 3 are the principal's office here. Last is the generator here, which you can see how the leaves are building in comparison to the principal's office. And also in the principles of this first picture, you can see the pests, how they breathe in the leaves. Thank you.
And there are some errors are there. For example, pH. You know, the way you have written both capital this one, that's not correct. Okay, P is lowercase. And experiment what you have said, you could have also done at your home. No, you would have had more samples. At your home also you could have done that in addition to these places, so that you would have understood what is in a different environment that would have behaved. Okay, so any one of you want to say anything up and down? Since Silva.
The comparative growth studies of green movement. Actually, today you presented, you understood the level of that presentation. You are not also happy with this presentation. Okay. Then another thing, that is study part, that is not properly when you put it in your slides. Then the picture, that all the images you should be labeled properly. You don't want it to tell first, people cannot understand which one you are choosing as a first. This other side people, they will think that is the first. This side, that's why the images should be. Madam, yes, that you should label properly. Then the experimental part, that is not appropriately used. That means you get the first introduction, then objective of that study, why you wanted to do this experiment. Another part, that is the experimental part, that how, where you are collecting the samples, what are the things, then where is the study area. All the things should be come in the systematic way. This is not the, you are not doing any effort in your slides actually. You should do properly. Another part, that what you mean by the heavy pollution. That's so one side you mentioned about that is heavy pollution. That means what? That everything will be unclear. Okay. That because your research part you won't find anything properly. Then research part also very big. That's why Sir is told to you that you please read some more literature and you study your subject properly, and to make it PPT also, please control your slides. So, what I noticed that you will think I will give to the other person. So, basically, you could tell like that. You could just handle the matter that the person talk. That's why you lost some time here. You have only minutes of presentation. 8 minutes only you have time. So, you have to divide among yourself properly. So, you need to tell them. Firstly, you will introduce yourself and you can start talking about it. So, no need to be like, I am just going to Sandra. Tell them. Okay? So, including the presentation and also when that results come, just interpret it properly. Now, it's almost properly. So, what is taking away from your study process? What do you want to tell? Only to the message from the study guidelines.
So from this study, we want to take away that this polluted environment shouldn't be the user. You should, for ideally use a condition that is clean. You should use a condition that may not have much stress. And one thing you have to absolutely make sure you regulate some of the water in the soil. Because if the field slightly goes off, they can cause further damage to plants. That's what our main takeaway from this is.
No, they see what is the suggestion is you do the literature review. Look at the literature, what kind of the environment this crop require. Okay, that is where it has to come in the introduction section. Okay, once you mention, okay, normally it is grown in this part of Karnataka, this is the kind of the soil required, this is the kind of the water requirement and all those things, you mention that. Then you highlight how your experiment is different from the conventional thing. Okay, and I want you to do the experiment at your home also, so that we have additional the samples. Okay, close the ribbon. Thank you very much.
Thanks.
Shri Guru Namaha. I am Vedha. I am Arun. So today we are going to be explaining our presentation our project on acclimatization of organisms in an aquatic ecosystem to a chemical environment. So as we know, India has the highest population in the world, and 70% of the population is involved in agriculture. So because of the growing demand, it is necessary for us to get higher yields. For this, fertilizers are very important, like they are very necessary. But at the same time, we have to make sure that they don't harm the environment. As we know, chemical fertilizers cause a lot of harm when they run off into aquatic bodies. So we want to understand the effects of these fertilizers when they run off into water bodies. So this is why we are doing this project. We also want to compare those effects to the effects when biofertilizers, which are better alternative for chemical fertilizers, their effects also we want to understand on water bodies, so that we can compare the two effects and then make a better choice. So our objective is to conduct a thorough examination of the effects of chemical fertilizers as well as biofertilizers on organisms in an aquatic ecosystem and compare their effects. We also want to highlight the advantages of biofertilizers in that way. So we took two study areas, which are Bannerghatta Lake, which is in Digri, and Kyalasanahalli Lake is also in Digri. This is a rejuvenated lake. Our procedure is that we took two large tubs in order to create an aquatic ecosystem in our biology lab. In that, we put soil samples from the same area, from the edge of our school playground. Both of them, as they are from the same area, they won't affect the results much. And then we put 10 liters of lake water. In tub 2, we put water from Kyalasanahalli Lake, and in tub 2, we put water from Bannerghatta Lake. Both of them have similar properties, so they won't make that much of a difference. So the atmospheric conditions was cold and presence of humid air as it was inside in our biology lab. The setup was inside, and then moderate sunlight came from the windows during mornings, and then we will also be looking at the effect on microbial communities by using agar plates. We have already prepared agar plates. Yesterday we prepared agar plates, and they are going to be checking the microbial communities first in the water before adding the fertilizers, and then in the water after adding fertilizers, and compare how they have changed, the communities have changed. So the organisms included in the study are semi-fighting fish, guppy, and microbial communities. In tub 1, we added guppies, wild guppies from Kyalasanahalli Lake, the same lake in which we took the water. We fed them with food like beta food. Both the fishes we fed with beta food. But the guppies actually showed more preference to beta breeding food more than beta food. So we put money plants in tub 1 as well as water lily in tub 1, and in tub 2 we put only money plants as we couldn't acquire another water lily. And by this way we created a lake-like ecosystem. We tried to create a lake-like ecosystem, and we left some time for the ecosystem to grow. And then after some time, after a month or so, in tub 1 we added Aqua Microbial Consortium, which is a biofertilizer with 5% concentration. And we have added urea with 5% concentration, which is a chemical fertilizer, to tub 2. Yesterday only we added it. So on the side we were also attempting to breed beta fishes because we wanted to see the effects on eggs as well. But we couldn't do it with wild fishes, as breeding wild fishes is a very difficult procedure. So we took domestic fishes, which is beta fish, which is quite easy to breed. The first attempt was actually a success, but the biology lab was not that safe of a space, so the nest was broken by outside force. So we'll be attempting to do it again. we did only a few tests as most of our time went in acquiring the materials and setting up the ecosystem. We also noticed indicators in the two tubs which indicated pollution before adding fertilizers only. And then we conducted calcium tests and silver nitrate tests, both the water samples in tub 1 as well as tub 2 before and after adding urea and AMC showed the presence of calcium and chloride ions. So we also did litmus tests, all that both before and after adding fertilizers, all the water samples were alkaline. But after adding AMC, in 2 days time only we noticed an increase in alkalinity.
I'm talking about the chemical stresses which were present in both the crops. So for both the crops, as she told, we have used the soil from the edge of our ground. Now that part of the ground in which the soil is taken, always has a flowering plant which leaves like a jam type of fruit, and that fruit often has its the roots of that plant always have it connected at that root. So we are expecting a lot of extra, expecting manure and the fertilizers which were used before also to help in the nutrient accumulation in that soil. So one of the major ones which we found in that whole thing was AMC. So as she said, AMC consortium, that was the most used because it was helping respond more profoundly to the helping our plants to grow. So for the female beta fish which I put in the tub, in the second tub, that of course that in the start had a very different behavioral adaptation. Like it was very stressed at the moment, but later on, as it got more accustomed to the place, it started growing more better and had better active response to the things. In the second tub, in the first tub, we had put wild guppies. Now the wild guppies, of course, they are resistant to almost every kind of ecosystem, so that's why they didn't have any problem in growing. So side by side, we were trying to also breed beta fishes and
So we have already conducted a few tests at our school. We are still conducting a few qualitative analysis and quantitative analysis. We are going to be giving the samples to external lab this weekend, and we are hoping to get the results soon and we'll update you on the results. So very slow changes are being observed as we have just added the fertilizers, and we expect fishes, the beta fishes and guppies, we are going to be adding guppies also in Tapu. We expect them to have more drastic changes due to urea, which we have added right now, chemical fertilizer, and all the work is in progress. We hope to update you soon. Thank you.
See, the attempt is good. It would have been better if we had done the water quality assessment of those lake also, lake water. You brought the lake water here and then you added chemicals, no?
We have the before samples as well. We have collected. We are going to give it for testing.
Not give it for testing. You should do it yourself in your
We did
lab.
some, yes.
Because the purpose of lake symposium is not to outsource the work. It has to be done in the respective institution itself.
We are doing it qualitative and quantitative analysis.
And then we are giving it is not appropriate terminology. Okay? Yeah, Cincy.
Your subject is different. Okay, it is fundamental. I want to get some clarity about what is the objective of your study. First of all. Another thing, why you use the money plant and water for your experiment. Okay. Then chemical stressor you are talking about. In that, please read some more literatures related to
So our objective is that
Yeah, I get it. I'm just given the points. Okay, please. You want to order? Okay.
Okay, you can do that. That's what she says. Okay.
If
Tulika,
you
you.
want to answer, you can answer.
so our objective is to provide an in-depth comparison between the effects of chemical fertilizers and biofertilizers when they run off into water bodies, nearby water bodies. And we want to make this aware to people so that they can make a better choice between those two and then help in saving, conserving the environment while at the same time reducing.
No, that is fair enough. What they are saying is, you should have the habitat condition assessment before you this one. All the four this one, no, in the lake water, what is the thing? Similarly here, when you got the water to the lab, what was the initial condition? After adding chemicals, fertilizer, what was the thing? Then you can say that what changes have happened in the organism. I am surprised with that behavioral study. You said that female, this one was, how did you make out that?
So because beetles are actually those fishes live in more tap water. So when she went in like fins, we can make out with the fins. Beta fishes when they stress their fins get all.
You should write that, okay? You say that how did you make out that? Because as a kid, we are also curious to know. Because we all know that how we can assess the stress. But if you can also mention that, that is a good lesson. Please do that, okay?
So one slide you have written in the conclusion part like fishes are stressed. Okay, so how did you understand it? It is stressed.
The double fishes were not stressed because it's the same habitat, like from the same lake water only we got. After MC also they did not show that much changes.
How many days is in this experiment now?
2 weeks once we added the fish, but it's been going for a week.
So you should also check whether is there any skin problems, colorations, because urea is actually ammonia and nitrate is urea ethanol also. Okay, so it is actually toxic to fishes. Okay, so you should continue this experiment for some more time and just check how the growth condition is. Are they getting any disease? Will they get die off? So if all that is there, then this will be very good experiment. Then how much quantity of water you are using?
You should write in that the samples chosen, what is the quantity of chemicals added, and then after adding, what is the water quality. You don't answer, you should do that so that the next presentation would be better than this. Okay, thank you very much. Yeah, one second. Sulika, answer.
Study area, you have got the screenshots from Google Maps, right? You can prepare the maps yourself and put them.
Shri Guru Namaha, good morning to one and all present here today. I am Ankur Desaiga, accompanied by my teammate Sudha Ramesh, and we welcome you to our presentation for round 1 of the IISC Lakes Symposium 2024. That is a comparative study on the different algae found in 3 different lakes in Karnataka and finding out whether these algae pollute or nourish the quality of the lake. So, as you can see here, this is our table of contents. It basically summarizes what we are going to cover in this PPT. And now I present it.
First, we are going to be showing the basic test, which is the tests taken to find the temperature of the lake, the pH level of the lake, and the alkalinity of the lake.
Next is the mineral testing, which tests for the amount of calcium, magnesium, and fluorides found in the lakes.
Next, we'll be moving on to the microbial structures found under the microscope when we tested the lake water.
Next is the dissolved oxygen, which evaluates the amount of oxygen dissolved in the lakes.
And last but not least, we have the gallery, which shows all the pictures of the microbes we found.
So, 3 lakes we have used for testing are, as you can see here, Sariki Lake, which is in Lakshmipura here, Puttenahari Lake, which is in Akkere, and Sariki Lake, which is in JP Nagar.
As you can see, in Puttenahari Lake, there are 2 sectors, the smaller one here and the large one. The smaller one here has a darker
tinge, and it is mainly used as a drainage and disposal area, which is where all the waste goes. Same thing can be observed in Saraki, but here it's a lot bigger due to the amount of population here. But in CK Paya Lake, there will in CK Paya Lake you will not be able to observe differentiation between sectors, because this lake is mainly used as a drainage lake due to the large amount of factories present around the lake.
So we chose the samples from the middle of the lakes, but if you give us the opportunity and time to continue with our project, we can take from different parts of the lake and even different other lakes. So now we're going to talk about the objective of the basic test. So as you can see, the three basic tests that we have chosen are alkalinity, average temperature and pH levels of the three lakes. So alkalinity helps in showing the level of a body of water's ability to neutralize acids and resist major pH changes. This reduces the chance of the water becoming acidic. Temperature determines and influences the biological activities and growth in the water bodies. So the temperature will influence the microbial structures found in the water bodies. So as you can see here, we have tested for temperature, pH and alkalinity in our school lab. So temperature of Putenali Lake is 27.0 degrees Celsius. The temperature of Sarki Lake is 22.0 degrees Celsius. And CK Pala Lake's temperature is 24.0 degrees Celsius.
So as you can see, Putanhali Lake and Sarki Lake and CK Paya Lake have a pH level of 9.1, 7.22 and 8.34 respectively.
The alkalinity of the three lakes are 480 milligrams per liter, 412 milligrams per liter and 390 milligrams per liter respectively. So from the observation of the basic test, we can say that 2 out of the 3 lakes have an average temperature of 24.3 degrees Celsius, with only Putenhali Lake being above average with 27 degrees Celsius.
2 out of the 3 lakes, which is Sarki Lake and CK Paya Lake, have an average pH level between 6 to 9, with only Putanhali Lake's pH level having higher than the average with a pH level of 9.1 due to its high alkalinity.
All 3 lakes have an alkalinity that is exceedingly above average of 20 to 200 milligrams per liter. But Putanhali, again, this lake has the most alkalinity with 480 milligrams per liter. So using all the information that we have gathered from the basic test, we can infer that Putenhali Lake has the most alkalinity and the highest pH as well as temperature.
The high temperature in Putanhali Lake may give rise to invasive microbes and species in the lake. And this is also not good because most of the fish species in Putanhali Lake are cold-blooded.
The high pH levels may also affect the fish and might be fatal for some of the fish. So now we're going to go to mineral testing. So the main objective of the primary objective of mineral testing is to determine whether the 3 minerals, magnesium, calcium and chloride, are present in the lake.
So as you can see, calcium and the chlorides are present in all 3 lakes, whereas now magnesium is present only in Putanhali and Sarki Lake, but it is not present in CK Paya Lake.
Observation of the previous slide. So calcium is present in all 3 lakes. Magnesium is present in only 2 of the 3 lakes. CK Paya having no magnesium. All lakes are taken for testing have chlorides in them. Using this, we can say Sarki's lake and Putanhali lake water can be considered as hard water. So now we move on to the microbial structures found. So we did this testing as well in our to ceratized algae, and we also found an abundance of microplastics and plant filaments. In Saraki Lake, we observed paramecium. We also observed a lot of plant filaments as well as a lot of microplastics. So in CK Palya Lake, we observed paramecium. We also observed Urina-like organisms as well as a microcystis colony of algae observed, and we also observed fungal spores. Next slide.
All of this, we are not sure that whatever we have found are what we think it is, but with further speculation, we might be able to pinpoint what it is.
So now we move on to the indicators in Putinvalli Lake. The ceratized algae is a very common colonial green algae found in lake water. It is an indicator of deteriorating water quality due to pollution. In sewage water, the ceratized algae is artificially added for oxygen and the breakdown of harmful chemicals. And also the abundance of microplastics is also an evident indicator of pollution in the lake. Next slide.
The paramecium was observed in Saraki Lake and is an indicator of pollution from biochemicals. Microplastics found in Saraki lakes are a valuable indicator of high levels of pollution.
As you can see, the paramecium, just as in Saraki, is an indicator of pollution caused by biochemicals. Now, the microcystis colonies. Finding microcystis colonies in lake waters is kind of like adding salt to your engine. First of all, it's very commonly searched for in lakes for high level for deteriorating lake water purity of the lake water. Also, these microcystis colonies are found in nutrient-rich and slow-moving waters. Although this may be a good sign telling that CK Palya Lake's waters are nutrient-rich, or they are also found in very slow-moving waters, which may give rise to breeding grounds for mosquitoes. Also, these colonies form dense blooms which are extremely harmful for humans if they come in contact with the skin or ingested somehow, with causing high amounts of injuries or rashes or ulcers in your digestive system and respiratory system. There are cases of in over 108 countries about diseases occurring due to microcystis colonies. Also, the high amount of fungal spores found in CK Palya Lake are also a surefire way to say that the snake has been notably polluted.
So now we move right along to the objective of dissolved oxygen. So dissolved oxygen, simply put, is nothing but the amount of oxygen present in any water body per milligram liter. Dissolved oxygen levels affect water quality, and if DO levels are lower than usual, it can indicate contaminated water. Average DO levels in rainwater are 6 to 8 milligrams per liter.
As you can see, Putinvalli Lake's dissolved oxygen is 1.7 milligrams of oxygen per liter and 2.3 milligrams of oxygen per liter is for Saraki Lake and 3.4 milligrams of oxygen per liter is for CK Palya Lake. All of this is below average.
So over here, as you can see, is the gallery of the algae that we found in Putinvalli Lake. As you can see, we found a couple of microplastics, we found microcystis colonies, we found plant fibers, and we also found some interesting types of algae, but however the identification is pending. But if you give us more time and the opportunity, we can definitely draw to a conclusion what the types of algae are.
As you can see, both of these are what is suspected to be a type of fungal spores.
So this we're not sure what it is, but we think it's a type of algae, but we're not 100% sure. So this is a video of the paramecium running in Saraki Lake.
It's a link.
No, definitely.
Internet is not. Net connect नहीं. It's Google Drive link.
continue the next. We will see that later. Okay.
Thank you.
See, actually there are a lot of time you said we are not sure and that. I think that statement you should avoid. You should do it and second thing is you should go through the literatures and understand the thing. There are many literatures related to these lakes and microalgae, etc. Okay, don't try to give the negative things saying that. Anyway, time is there. The symposium is in October. You try to improve that and come with a good quality project, okay?
Yes, sir.
So, Cincy?
So only 45 lectures are available in the lab, so that's why we are able to identify exactly.
That's okay, you talk to your school. They will talk to other institution and arrange for you. And there are some labs are there nearby where students can go and do this one, okay? You might have seen some of the people have started the labs outside, not the commercial one. For the education only they have started, okay?
We definitely do, sir.
I have some suggestions. I don't want to give an answer. I'm just given a point.
Next also
You should
PSBB.
write that part. But you should keep it in your mind and you should improve your slides also. Okay?
Yes, sir.
First one, that mg per liter. The alkalinity that mg per liter, that is the SI unit is there. You should use that. I don't want you to write as a mg per liter or whatever. Okay?
Yes.
With the values we have to write mg per liter. That wise you should start on you should read some more literature and publish the articles and all. Okay. Then another thing that you were that the observation of basic testing in that second bullet, that pH value, that Kuttenhalli Lake pH value is 9.1. That pH, that P is small and H should be capital. Okay. Always you should be keep it in your mind. There that second bullet that is wrong. Then what is the time that your sampling collection time? That you put salinity and all the things. You are talking about the temperature. So that time is very important. No?
See,
That temperature-
whether you collected in the morning or afternoon, you should mention that. What time, whether 7 o'clock, 8 o'clock.
We collected around the afternoon. We are not sure about the exact time.
See, there is a protocol, for all this thing, one has to do before sun rises. You have to keep that in mind. That's where you should read the literature. We have put a document online, no? You should read that, okay?
Yes, sir. Yes, sir.
That is very important thing, that because we are talking about the temperature, but it is you have everything like microbial activities and all things. So the temperature time is very important. Okay. So another thing, the microbial structure you are writing, that is not the correct way. Okay. Then you are asking, put it as a algae also is there, then paramecium, everything it will be there. Okay. That is not the correct way. That heading also should be changed. Then the abundance of microplastics. Most of the things you are talking about the microplastics. So you have in your sampling collection, you saw that anywhere in the water, that static lake or wherever lake, you saw that the plastics are floating in the water.
Yes, sir.
Then you should take that picture and put it in there also. Another one more thing, that microplastic, that image also you showed. What experiment you did for find out that microplastic? Or simply you just put it in that water sample and put it under the microscope?
No, we've taken the water sample from the lake and what we observed.
Yeah, no. Microplastic, we wanted to find out. No, that is the procedure is there. Different procedure is there. You should do that now, then you can tell like that. Okay, you didn't do that procedure and do this now, then you cannot tell like that. Okay, but you can tell the thing, you can show the picture that this floating
See, actually dissolved oxygen has to be done in the field itself. When you collect, no, you have to do the analysis there only. Not take it to the lab, bring it to the lab, okay?
So I think the values are not proper. Just do once again. You only go to the next one, okay? And what about the algae species beyond that? Or the algae species, you cannot show the pictures at all, and you cannot label properly. So you should label properly, and so you do microsystems, but I don't see in the quality itself. I showed that, I saw that. But I'm surprised that you didn't see that with microsystems they only take. So again, you have microscope test tool, right?
Yeah.
So again, you look at all the lake water samples, and again they take pictures of whatever you have possible. You can just take pictures and try to identify, because not only sentences will be there in that water. So if you add more, then it will be more better. Then we need. So try to identify those species, and also you can take picture parameters also. You just put a photograph and label all the species that you got. Okay. Sometimes that video won't, we won't get the time to within 8 minutes. Now you got 10 minutes. But in the advance you won't get the time, only 8 minutes. In that time, so it's better. Okay, I got videos. You put a photograph. Like photo with a label. That is species now. That is should be in Italians. Not the species that only genus name. Then it will be. So it is like if you realize there's some what it indicates pollution like that. It's there. So you refer to some more literatures and write that under the indicators.
My name is Aruna and this is Ritwik. Today we will be taking you through the detailed journey of an analysis of the growth of Bannerghatta, that is TVRAM, or commonly known as blue in India, across various soil types with nutrient variations under peculiar variations. So India is a global powerhouse for pulse production, including legumes like mustard, mungpeel, urad, and many. So leguminous plants are known for their nitrogen-fixing capabilities, which not only helps in self-sustenance but also enriches the soil for subsequent crops. Now the reason we have chosen Indra as our species for the experiment is because it has a relatively lesser, shorter growth cycle and a higher nutrient sensitivity. Now further, India is a country that's rapidly shifting to urban ecosystems. So we felt understanding and analyzing the adaptability of these plants to the varying ecosystems, environmental factors and nutritional factors is more crucial. So this study aims to observe the interplay between various environmental factors, nutritional factors, plant growth, and the varying ecosystems of India. So the absence of preliminary soil testing is intentional because we wish to understand how TVRAM responds and adapts to the variable unknown nutritional factors in the soil. And we believe the results and the experiments made from this project would serve as a baseline data for us to conduct future investigations that could give us an insight into analyzing and arriving at optimal nutrient combinations and nutritional strategies for farmers all across India to take on. So our ultimate aim of this project is to equip our Indian farmers with a good toolkit and an advanced nutritional strategy that would allow them to give a bumper harvest every year after. Now coming to the methodology behind the experiment, a thorough survey of the locations across our school was conducted. So we found out 3 different sites that could resemble Bangalore's varying ecosystem to the maximum. The first one is a pristine natural environment, which is our school garden, that would resemble the natural ecosystem of Bengaluru. Second is a concrete enclosure, that is our school office, which we specifically chose this spot to highlight the inefficiency of new makeshift gardens that one could observe between 2 houses in Bannerghatta. This condition situation is only going to get worse. So this controlled environment with higher humidity is a place that would resemble the urban jungle of the urban ecosystem of Bangalore. And the third one is the polluted area, that's the Genset PSBB area of our school, which would allow us to highlight on the environmental stress faced in industrial areas. Now coming to the variables in the experiment, we chose 4 major soils that we could observe over the entire nation, namely A, black soil, B, red soil, C, sand, and D, cocoa peat. We also used a unique identification code for each species sample. So as you can see, this is the data matrix that we have studied. Further, now I will be explaining about the methodology behind the experiment. First, we started off planting seeds in 12 identical jars of 150 cubic centimeters of different soils each, and then we watered them regularly using tap water, some on a regular basis and some according to the individual requirement of each soil. And we collected data, some on a regular basis, a daily basis, and some on a weekly basis again, like stem color, lamina color, lamina width development, and other growth parameters like height. Now we will be explaining about our findings from this experiment.
So we can observe in the urban environment that the growth, so we can observe the urban ecosystem that the growth was the worst. This is due to the high humidity and the lack of adequate sunlight. The plants could not grow. Next slide. So now in the natural environment, we could observe the best growth. This is in that graph there. We can observe the best growth, but however, there's an issue of a lot of pests biting the plants. So if this issue of the pest was taken care of, this will lead to a bumper harvest very quickly. Next slide. Now in the polluted environment, we could not see any issue in the growth until day 30. A white residue was also observed on all of the leaves. Now we'll talk about the different soils. So first about sand. In sand, the highest growth is observed. This is as 45% of mugwort growth is done in Rajasthan, a place with mostly sandy soil. And also next about cocoa peat. Cocoa peat is a supplement that provides good aeration and also good drainage for the plant. However, it does not have much nutrient value and should not be used as a primary soil. It will be used in combination with sand to provide a good harvest. Next about black soil. The black soil contains a lot of calcium to help with which should help with the growth of the plants. Next about red soil. The red soil in the case of the urban ecosystem, the red soil was the only one that could survive until day 20, while the other ones, the other plants could not survive until day 15. We need to do further research to understand why this was. Yeah.
So basically, one speculation that we came with was the preliminary pH test that we conducted for red soil. It had a slightly acidic pH of 6.5. So we are intending to do further studies on this topic. So we believe maybe the 6.5 pH could be the reason for red soil surviving the longest in the urban jungle environment. And also for soil nutrition level tests, we have given this 12 jar samples to the biocenter at Bangalore. we are okay in the future we are planning to conduct some preliminary tests for calcium, magnesium, chloride, and everything in our school itself. But however, through a thorough nutrient profile analysis, we thought we lacked the equipment, and only these lab places had the appropriate equipment for it. And these results are expected to arrive around the end of this month. And one more thing to summarize, again I'm not using the word conclude because we believe research is a continuous process. To summarize, during this entire project, we came with a lot of difficulties. Although it was only 12 jar samples, we had to safeguard the plants, the saplings from being trampled over by kindergarten kids, and we had to safeguard them from pest infestation and other environmental factors, even stray dogs and cats within the school campus. And now we salute the Indian farmers who, of course, face unimaginable difficulties when they conduct this at a much larger scale. So I know right now all these findings from this experiment are only the baseline data for our future studies, which will include correlation of these plant growth observed with specific soil nutrient profile stages. So our main aim is again to provide farmers with a toolkit and specific nutrient strategies for giving bumper yields, and we intend to set up a better future for our Indian farmers. Thank you.
In what way your experiment was different from there was a similar experiment, no?
Sir, so actually we were originally a team of 5, and then we heard information from the IISC that maximum of 3 members were needed. So we separated our topics so that our future findings would be related to nutrient profiling, and they will concentrate more on pollution and pest infestation. So that's why
But
we are
that didn't
waiting.
come out that way. We didn't have clarity on that. Anyway, you can refine it, and also as I said, you should have the literatures this one, and mention about what are the kinds of soil across the state, and what kind of crops are grown in those things, and what is the kind of favorable the climate, favorable nutrient status in the soil required for that particular crop. Then talk about your experiment, in what way that is better. Some part you put it very well, but you need to refine it. Sincere.
I have some suggestions. First one, that Vigna radiator. You should give that plant the characteristics. Okay. So it will be that without knowing the plant characteristics, how we can tell the plant growth and all that. That is the another. Basically, we wanted to know, we wanted to see, this scientific name you are putting. Some people don't know the plant growth. The conference donor, that different people are going, different subject also they are on. So you should give the introduction about the Vigna radiata, that species. Then many other literature is there also. You should bring that part also in your introduction. Then so recently I looked into the research paper of the Century University. In that we saw about how the same plant they had conducted a similar research and found out that 60% of phosphorus and 30% of sulfur is the best combination for growing wheat plant. So in our further experiments, we intend to do something like that. Once our lab procedures are the results reaches back, we intend to find out something like that. And
No, you have to be careful in reading the literatures. Okay, this is not high nutrient, is not necessarily will give you higher phosphorus, and what you mentioned is a high nutrient status. please make sure that, okay? There is Agriculture Science University is there. No? You can go to the library and refer the literatures, okay? For the Indian condition, you need to look at it, okay?
Then another point that is your objective that green plants responds to unknown factors. I didn't get that unknown factors.
So can I give an example?
No, whatever point you are adding into your slides now, that everything should be come to the result part. Your objective, that's why you please answer that. Give the very clear objective of your study. Okay? That's based only that your introduction, that based only that your materials and methods, that based only your that your research and discussion. I'm just giving the point. So that we can add whatever we know that we can add, but based on your project, you should be very clear of that your objective of the study. Then introduction and discussion. Okay. Please start with the points.
Okay, thank you.
Thank you. Thank you so much.
Graph, you know, put on the graphs. The y-axis is not clear. What the y-axis means. Y-axis title. What the y-axis means. X-axis we have put yes. Y-axis is not clear.
Yes.
And one more thing, in the references you have mentioned Wikipedia. Wikipedia is not a reference. It is a crowdsourcing platform. You should look for original sources for your information and
Thank you.
Project is headed like acclimatization of organisms and plants in an aquatic ecosystem to a chemical environment, in which we are going to do about plants. Myself, Diksha of class 9, and since
Sujay from class 9.
So, introduction. Chemical fertilizers have been used on a large scale in India because we 70% of our population are involved in agriculture-based practices to earn their living, and but then excess of these fertilizers can cause damage to various elements of the environment. Over here, as the head of IISC suggests, it is about lakes. So we are going to explain how the fertilizers which drain into the water bodies can affect the aquatic plants which exist in the area. So our objective is to analyze how chemical fertilizers can affect plant growth and quality in aquatic ecosystems. Additionally, it will also explore the benefits of using biofertilizers. That is, we have used Alpha Microbial Consortium (AMC) in our research. So how that is also helping in reducing the chances of the environment getting polluted. So our study area, we have two tubs. One is from the lake Bannerghatta, which is in Germany. It is spread over 700 acres, and the water holding capacity is about 300 acres. 300 acres there.
So now we have taken for the samples water lily in tub 1. So to take it as a sample, we took actually a water lily as a sample from the artificial pond near the junior block of our school, and it wasn't blooming, though it was being grown in that artificial pond for 13 to 14 years. It wasn't blooming wholly, but after we took it and transferred it to our tub, it started blooming. So there are few reasons that it started blooming in just 2 days. It's because that it had its root couldn't spread more in that pond as it was being kept in a container. And the second reason is that we humans are giving it more carbon dioxide and it has moderate sunlight. That's it.
The materials and the methods we used two tubs, namely tub 1 and tub 2. We used the biofertilizer Alpha Microbial Consortium and the chemical fertilizer urea. And for collecting the samples, we have gone to the lake and collected the sample from the middle and from the edges of the lake. And in the atmospheric conditions in the lake was mostly humid, and it was very the lake above in the middle of the lake it was very cold, and on the edges of the lake it was quite warmer because there was the presence of a lot of trees around. And the place where we have done our research is in the biology lab, and what is connected is from Bannerghatta Lake and Callison Facility. So we have done a few simple tests that can be conducted in our lab, in our chemistry and biology lab. That is the fluoride test and the calcium test. But we could not exactly acquire the exact quantity of that because we needed a chemical called ADTA for that. So we were going to acquire that when given opportunity and time. Procedure.
So we took 2 tubs. We took the soil from the edge of our playground, and because the soil was very normal, very soft, and so that is why we took it. We added it to the boat tub, and the plant, the root, and overall.
And the purple water lily as acquired from the artificial pond, and the money plant was also acquired from school premises. Money plant, it is very easy to acquire because even the stem can be put into the plant and it will start spreading. So the calcium test was done in the following ways. We took water, acetic acid, ammonium oxalate, and we mixed them together with our sample and if it produces a white shade, it notifies the presence of calcium. We would notice that tub 2 had a higher calcium quantity than tub 1, and the chloride test, that is also known as the silver nitrate test, was also conducted at school. It was done by along with the addition of the sample water, potassium nitric acid, the silver nitrate AgMMA, and we noticed that more ions of chloride was present in tub 1 than in tub 2. We also took the pH test and the litmus paper test. We found out that when we had first got the control samples, we were close to 7 in pH, and when we had after the addition of EMC, we noticed that it had acquired a higher pH, that is around like 8 to 9, and at home we had conducted a simple water hardness test, that is done by mixing water and liquid soap like dishwashing soap if the water sample is still murky after settling for some time, it notifies that the water is hard, and if it has a ladle on the top and the water is clear, it notifies that the water is softer in nature. And all the 3 samples, that is the Bannerghatta Lake control sample, the Callison Lake control sample, and the AMC water sample, 3 of them had a lot of hardness in them. This can also notify that magnesium is present because magnesium also determines the hardness of water. So the concentration in which we have added the biofertilizer and the urea is 5% and 5% each, because we have noticed that we have acquired the biofertilizer from a local lab and also a research center, it is bio center. So we are told that most of the farmers have been using this lately to compensate for chemical fertilizers, and urea was added to tub 2 in 5% concentration. That was the average about which normally farmers had, and we were also frequently incorporating gaps so that the plant roots start to grow.
So after adding the AMC, we noticed that the water lily started rotting up, and we also noticed that the money plant was also rotting.
So the purple water lily Bannerghatta was 27.5 centimeter in length when it got the first stem with the part, and the money plant was in tub. We have it in 2 tubs. So in the first tub it was 73 centimeter, and in the second tub it was 54 centimeter. So nutrient requirement of water lily.
The nutrient required for the water lily, the optimal soil for cultivating water is rich in organic matter, and these plants require a plant space of 1 meter by 1 meter. The water lilies are typically nourished with lily ducts or slow release aquatic fertilizers in our project. We utilize Arca microbial Cosmodium AMC for feeding the water lilies.
We use money plants do not typically need any fertilizer or anything in specific quantities. It can grow even in the smallest areas, and it mainly also needs nitrogen and phosphorus. So we have utilized urea and Manda and AMC. So the water lily, as we had already mentioned, before the addition of the AMC, it was fresh. It had lush green leaves. Then after the addition of AMC, it started to rot. This was probably because of there was a higher concentration of the chemicals which was not there, and also as mentioned, the water lily had a more space to spread out, and there was the air quality over here in the biolab and the air quality in the outside open artificial pond was very different, because over there the buses used to pass and the air is very polluted, and in the biolab the carbon dioxide released by us is sufficient. The money plant also started to rot when we had introduced it at the first. In tub 1, it had become yellow and wilted. That also signified that iron was not present. And in tub 2, it remained the same color. It was lush and green. It had a lot of leaves. So in conclusion, we have to say that chemical fertilizers have created a very different environment change in the tubs, and they have also notified that if used in inadequate quantity or in inadequate quantity, they will affect the growth of the plant.
You took longer time than and I think the suggestion is same as what we gave earlier for the earlier presentation. Okay, you need to have the literature to highlight why this experiment was conducted. Okay.
So
You can come up with that, okay? Since Suva will tell you.
Same pronoun, same sentence, same language.
Are they both concepts at the same class? No, actually they were all together. Same class is at the same class. They were all the same.
No, they should have put in effort. I think the children who have prepared this slide should have used this. The others should have prepared new set of this one.
So the experiments, everything was same. So only around the time of submitting the IISC got more time to prepare what it was too late for them to start something new, so we. So can you change this? They are the same category itself.
You are in 9th standard now. But two teams will present one half only. That is the problem. Then judges also same judges.
If they are different standards, then we could have like.
It will be so. One thing
We should come this.
can change the subject.
Can we test any other parameter? Instead of chloride and
Another thing. Why you wanted to do this study?
To provide like idea to the farmers that using chemical fertilizers is very harmful because there are
In the money plant and the water lily, how you can
Tulika.
already
Net.
in the ecosystem it was already there in the lake. Money plant and water lily were there, so hence we had added.
Then what information you want to give to your audience?
That the long-term use of chemical biofertilizers can and the short-term use of chemical fertilizers can be taken into account. And if you want to have higher yields or if you want to have more organic ways of farming, you can adapt into the
Okay, here there are teachers, students and teacher corners are there. Okay, you can refer that. See, in this, before there's an orientation program, what we did, all those details are available here. You know, the presentations, what we did, school wise we have put it so that you can go through that. Okay, suppose if you take this, BGS you had come, no? Each one of us, our presentation videos are available. You can have a look at it. Then after this one, you know, the evaluation, you know, we are going to put what you have presented in this section. Okay, similar to what we have done for the other school. Okay, that is not coming. Lake. Okay, see, we have done in Aromira, that presentations we have put. These are all the presentation what children have done. Okay, their slides as well as the son. Okay, this is as far as the conference is one. And this is the reference material which I said. See, here there is a environmental monitoring. The children who are working on water quality etc. can refer that. Okay, and there are other informations are also there. Information on plants, butterflies, birds, etc. You can refer this section, okay? And if you want to see the other details, you can go to our website. You know, there is the research group, our research papers are also available here. You know, the research paper sections here and here will give you the theme-wise research paper. See, these are all related to the lakes. You can refer that, okay? Now with this, I request Tulika to talk about the maps, okay?
Okay. So, who have not implemented and who have not seen, for them I will give you a small demo and it is already there on the website also, so you can refer it to that also. Okay. First is how to work in a field. Okay. First point is we always go to the field in a group. Okay. We never go alone. So in the field, one of us will be taking the point, one of us will be noting down all the points that surroundings, what is the climate, what is the temperature, all those things, what is the date, what is it raining, is it a sunny day, everything you have to note down, because you have done the water quality assessment, you have done the soil quality analysis. So the quality of the water, soil, all those depends on the surrounding environment, correct? Correct or no?
Yes, ma'am.
I can't hear you.
Yes, ma'am.
Okay. Can you all hear me?
Yes, ma'am.
Okay. So now we have gone to the field. Now we have to take some photos. First, our priority is taking photos. Anna? Okay. So now, to take the photos, first thing we need is GPS. All of you know this device? What is this? How many of you have this device? Yeah. How many of you have this device? Anyone have? No? But I'll tell you, all of you have this. Do you have this?
Yes.
All of you have?
Yes, ma'am.
How many? I want to see hand. So all of you have mobile phones, right?
Yes.
So in your mobile phones, your parents has, someone in your home has, your friend has, right? So in that mobile phone, your GPS is already there. So now all of you have GPS.
Yes, ma'am.
Now you all can take the field photographs
Yes,
with
ma'am.
coordinates. Okay. Now first is our priority is taking photo with the location. Okay. First thing is GPS map camera. You all note down this. GPS map camera. Using this, what is the advantage? Works simply like the phone camera. Whatever phone you are using, normal camera that you use, it works simply like that. The main benefit is, can you see that photo I have taken? The benefit is we will get this information. Which location I have taken the photo, which time, which date. All the things we will get from this app. Okay, only one condition, you will have to require the net connectivity. You all have that?
Yes, ma'am.
Yes or no?
Yes, ma'am.
I want louder answer.
Yes, ma'am.
Okay. Next, one is, see, I have taken a mango orchard. Can you all see the peacock?
Yes.
Can you see which date I have taken it? Which time?
Yes, ma'am.
So now you all know what is the condition of the ground, which date, all the things you know, right?
Yes, ma'am.
But just seeing the photo. This is what I was telling you. When you go to the field, you take the photo using this app, so that all the information come in a single picture. Okay? This is also a similar kind of thing. This is a water bird. Then next application is Google Earth. This also you can install in your phone, and using this you can take the photos in points or the location in your field. Okay? Now some of you have not made your maps. I really want all of you to make your own maps. Not take screenshots, not take the maps from Google search. Just make your own maps. Yes? No, you will make nice, full resolution, high resolution, good maps. Okay? So how many of you promise me to make your own maps? So I'll teach you. Okay. So it is not tough. It is very interesting, to be honest. So now we will see something called Google Earth Pro that you can install in your laptop or your desktop. Okay? This will help us in many ways. We can create a map. We can, like I said, you can go back to the 1980s and see what was the condition. Some of you have done the temporary analysis, so that you can see what was the condition then and what is the condition now. Here you can measure the area, the distance, all that things you can do. Okay? Then this is the installation process. You have to go to Google and search for Google Earth Pro. What do you have to search for?
Google Earth Pro.
Louder.
Google Earth Pro.
Okay. Now when you go to this website, this option will come. Download Earth Pro on desktop. When you download it, you will get directly zoomed in to wherever you are. Okay. Now I'll give you a small tour of Google Earth Pro. So there is on the top menu bar. Okay. Just beside menu bar there is toolbar. Okay. Then there is search bar that works simply like Google search. You can search for any location it will show you the location and you can go to that location. Okay. Then list of the places, the points, lines, whatever you add, the locations that will come in the list of your places. And there are multiple base layers which can show you the road network, the places, names, all the things will come in the base layers. Okay? Now tell me what is point, line and polygon. Yes.
Point is the area particularly.
Okay. You? Okay. Anyone else? Yes.
Something that we use to mark location.
Okay. What is line?
Road connecting to road.
Okay. And?
Mark roads.
Okay. And anything else? Rivers. Okay.
Railway lines.
Railway lines. Okay. What is polygon?
Area.
Area what?
Anything.
Any area.
My area?
Any lake.
Area of any figure.
Okay. Figure?
Any lake or place.
Area of a particular place.
Particular place. Like if I want to mark the campus. If I want to mark the campus. Okay. If I want to mark myself. Point. Okay. If I want to mark the way from here to my home. Very good. So now we know how to make point, line and polygon. So now we will see how to take this in Google Earth Pro. Because we need to make the maps, right? So to make a point, first thing is, this is the toolbar. You remember toolbar? Yes. So here, this is the option. You can see this yellow pin point. It is called add place mark. Okay. When you click on this add place mark, now you will get this trace here. So either you have the latitude longitude when you are taking the photo. Remember that value you can put here. Latitude and longitude. You can place the values of latitude and longitude here, okay, that you have taken from GPS map camera, or either you can just shift your point to the location that you have taken the point from. If you do not have the latitude longitude, then the latitude longitude will automatically come in that place. Okay, done. Now you all know how to take a point location. Now after you have done this, you can give the name here. Also you can change the color from here. Okay, but to mark a particular location, you need to give it a particular symbol. Okay, to give it a particular symbol, there are many options of symbols. So many are there. So next, this white and black thing is here in the middle. This one we use to mark a particular location of a point. Okay, the samples you are taking from some lakes, all the things you can mark using this particular symbol. Okay. See, I have taken the photos of macrophiles. Can you all see? So now the names are also there and the location, their latitude and longitude where I have spotted them. Okay. Next. So you can all next do the point location. You all know how to do that. Point location is done. Next is line. Same way you can mark the line. Like see here, I have marked the line here. I have marked the road. Okay. That you can do using this option. Add path. Okay. First, second, third option. Okay. Here also you can change the color and click on OK. It will be saved. Okay. Next. Here you can also calculate the length. And there are different units of measurement. Appropriate thing you should select. If I am marking my distance from my home to here, I will use kilometer, right? If I am marking the distance from here to there, I will be using meter, not centimeter. Correct? Okay, next. Next. Now finally the polygon or area that you are marking. Okay? That is the second option called add polygon. Simple? Add polygon. So here we can see I have the perimeter. Someone showed perimeter also we can mark, and also the area. Zoom out. Here also we can give the name and change the color. Okay, and then click on OK. It will be saved. Next. Now all of you missed this part. To mark a particular land use or land feature, you have to focus which land use or land feature you are focusing on. Suppose I am focusing on a building or a school campus. That should be marked by red color. Okay. If I am marking a agricultural land, that should be marked by yellow color. If I am marking a water body or lake, that should be marked by blue color. Okay. And if I am marking a like park or a vegetation land, that should be marked by green color. Okay. You all understood the color thing? While I am drawing a scenery, बनाते थे ना वो घर, घर के बगल में नारियल के पेड़, फिर आगे से नदी बहता हुआ, तो नदी का कलर हम गुलाबी थोड़ी देते हैं, नीला ही तो करते हैं ना. ऐसे ही the forest should be green color, buildings red color, agricultural lands yellow, and the water bodies blue. All done? No confusion? Okay. Now here also you can do the calculation of area and the perimeter according to your unit of measurement. ठीक है? Next. 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 So here this is the historical maps where you can go back in time and see what was the condition before. It works like the time
Give an
machine.
example of that. One of the student wanted that Arakere lake, no?
Second last presentation.
Arakere lake,
है ना?
that.
Second, who was that? Yeah, you did that. No. You guys did. Yes.
Historical thing.
So, like you saw na, the earlier image and the current image, they show 2011 and the current time. Next. So here I have given an example of 2011 and 2023. See how you can see the changes in these regions. How these buildings has come up more, right? Next. See, this is the image of 2010. How it has changed in 2022. Like that, when you are doing, you are saying this region has improved or this region has deteriorated. This can be based on temporal analysis only, right? So for that you will require some previous time knowledge also. So for that this thing, this historical mapping will help you. Okay, next. Now you have taken the points, you have taken the lines, you have taken the polygons. Now it is time to make our final map. Okay, for that on the top, on the toolbar, you will have this option called save image. Here, this one. Okay, what is this called? Save image. You click on this, these three options will come first. Okay, and these things will also come automatically. So first here is the title. What does the map show? ठीक है? हम जब अपने फोटो लगाते हैं, नीचे से वो कैप्शन नहीं डालते हैं ना? So that shows what is there in that photo, है ना? Simple. So like that. This is the title. Then this side legend will come. Legend will show what are the things कि उस फोटो में मैंने लाल कलर से क्या लगाया है, ब्लू कलर से क्या लगाया है. That will come in the legend. You are understanding Hindi, right? No problem. Okay. So title will show what is the thing in the map. Okay. Like if I have put my image, I will put my name. This is my image. Okay. Suppose I have marked a lake by blue color. Then what does that blue color show? That I will show in my legend. Okay. Now I have added all these things. Now it is time to save our map. Here, save image option will come. You have to click on this. It will be saved in your desktop. Okay, as an image, JPEG, PNG, whatever the format. Next. Next. Now, the final output, your map should look like this. Okay. Now, to finally understand what components should be there in the map. Next. 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 Back. So there are 5 components should be there in your map. Okay. First one is the title. Okay. Second one is the legend. This which way is the north? If I tilt this map this way, so north arrow will be tilted. है ना? So this will show if I have tilted my map or not. Okay. For that, north arrow is also important. All of you can see this north arrow.
Yes, ma'am. Yes, ma'am.
क्या हो गया यार? खाना नहीं खाया क्या? तो जोर से बोलो ना.
ये है स्केल, ठीक है। ये शो करता है कि दिस विल शो हाउ मच एरिया वी आर रिप्रेजेंटिंग इन द मैप, ओके। हो गए 5 कंपोनेंट। व्हाट आर द 5 कंपोनेंट्स? टाइटल, legend, not arrow, scale, fifth map, correct. So what are the 5 components should be there in your map? Title, legend, not arrow, scale, map. Again, title, legend, not arrow, scale, map. You won't forget, right?
Yes, ma'am.
All the presenters that are here, I want to see your maps with proper 5 components in next signature. Yes or no?
Yes, ma'am.
Do you promise?
Yes, ma'am.